Yeah, so thank you for giving me this opportunity to share with you work which we have been doing through the PRISM Drug Resistance Project. And I'll basically be talking about emergency and spread of artemisinin resistance in Uganda. And this is actually work which was done by a lot of people. I'm just presenting many people who contributed towards this work. So like I said, my talk will basically be focusing on parasite resistance, and this will mainly be on artemisinin resistance. As you may know that ACTs are actually key for malaria management, but one of the major problems right now is that resistance to artemisinin, which is a key component, is actually threatening their importance for malaria management. And everyone in malaria community is concerned that this could actually worsen the challenges of malaria control that we're facing right now, and more so in Africa, where more than 95% 95% of malaria cases and deaths actually do occur. Artemisinin resistance itself actually involves basically delayed clearance of parasites after they have been exposed to artemisinin components. And basically, it is not the classical antimalarial drug resistance because the parasites actually do respond to the drugs eventually. Only that there is an experience where there is delayed response, which mostly affects the early ring stages. And this phenotype emerged early in Cambodia, that was around, I think, 2000, and now it is widespread in Southeast Asia. And phenotypically, artemisinin resistance is characterized by one in vivo clearance half-life of around 5 hours or more, or day 3 parasite detection after initiation of ACT therapy. And in vitro, this phenotype has been detected through an assay called ring state survival assay, which involves higher levels of parasite survival when they are exposed to high dose of DHA for a very short time. And in terms of genotypes, it is associated with genetic polymorphisms in the Plasmodium falciparum catch protein, which I'll be referring to as the K13 gene. So in terms of genotypes, there are more than 100 different K13 polymorphisms which have been detected. And not all of them are associated with the artemisinin delayed clearance. On the right-hand side, the figure above the actual shows one of the reports from Ashley, which shows on the x-axis where you see parasite clearance, on the y-axis where you see the different genetic mutations which are seen along the K13 gene. And the parasites which are in red dot are the ones which have mutations along the propeller domain, and then the blue are the ones which have mutations outside the propeller domain. And then, of course, the first one shows the wild-type parasite. So what you see actually in that figure shows that it's only the parasites with mutations in the propeller domain that are having clearance half-life of about 5 hours, but those outside actually do not. And out of those mutations associated with delayed clearance, approximately around 21 of them are now being clearly documented by WHO, and 3 of them have been reported in Uganda and Rwanda, and those are the ones we have highlighted in red on the lower figure you see here. So as like many malaria endemic countries, Uganda has also been faced with challenges of particularly antimalarial drug resistance, and as a result, there have been changes in malaria treatment policy over time. Just to give you a glimpse into that, in 2000, chloroquine was the main drug for malaria management. Because of resistance, this was shifted to combining chloroquine with sulfadoxine pyrimethamine, although both of them were also having high level resistance, but there was hope that combining the two would redeem some efficacy, but that didn't work. So therefore, in 2006, the National Malaria Control Program in Uganda actually adopted ACTs for management of malaria. And since then, for more than 1.5 decades now, Uganda has been using mainly artemether-lumefantrine as the first line for malaria management. And for the over 1.5 decade use of AL. So the key question is, what is the impact of this long period of AL use on local parasite population? And specifically, we're referring to the parasite composition in terms of the resistance genotypes in the parasite population. And a lot of people or research groups in Africa have also been interested in that kind of question. And on the map here, what you see is actually different studies that have been undertaken to try to understand what are the risks or what are the prevalences of the key mutations that may actually drive artemisinin resistance. And the actual number of studies that you can see in the map here that have been trying to do that. But until 2015, relevant PFK13 mutations were rare in Africa, but it was recently, like in 2018, there when more of these polymorphisms were seen. And in a review that was done by a group published in 2021, they actually found that 4 African countries reported around 6 of the validated K13 mutations. By validated, we mean those which already know are fully associated with artemisinin resistance. So those were 6 of them reported in 4 countries. Meanwhile, also in Uganda, through the PRISM drug resistance project, which is a project under the International Center of Excellence for Malaria Research, Uganda program, an NIH-funded program. In this project, the key objective was also to assess the temporal and spatial prevalence of antimalarial drug resistance markers in Uganda. And the key approach was to go into different study sites. This involved 16 health facilities, as you can see in the map. And the ones which are highlighted green are the first one where the study started from. That was in 2016, and those were 10 sites. And then later in 2018, the number of sites was increased to 16. And the new ones that we added are the ones which are marked yellow. And specifically at each of those sites, what we did was to collect dry blood spots from 100 participants who are diagnosed with malaria based on either malaria rapid diagnostics test or microscopy. And all the samples that were collected from 2016 until 2020 were genotyped using different approaches. And along the way, we used the Luminex platform to look for polymorphisms in the genes, which we already know markers for. We used the Sanger sequencing specifically for the PFK13 gene because we know the number of polymorphisms that might be of importance in that gene. And then currently we are using the molecular inversion probe-based next-generation sequencing approach to look at a number of polymorphic plasmodium genes that we know are important for antimalarial drug resistance. And of course, we also use qPCR at some point to look at copy number variation for genes like the plasmepsin-2 and PFMD1. That was at the start of the project. And we did some conventional PCRs for microcellular because we're also interested in looking at diversity of some parasites of interest. And like I said, the study started early in 2016 and is still ongoing. So far for the period we have undertaken this study, one of the key results that we observed is that there are first 2 important PFK13 polymorphisms that were detected early in 2016 at very low prevalences. These include the 469Y, 675V, and these 2 polymorphisms, like I said earlier, are being known to be associated with artemisinin delayed clearance in Southeast Asia. So as we continued with surveillance from 2016 to 2020, key point here is we kept seeing the prevalence of these 2 polymorphisms, which are shown in the first 2 rows of map increasing. So by 2020, we're actually talking of prevalence increase from around less than 10% in 2016 to nearly 50% in 2020, which is really a substantial increase in prevalence, which clearly shows that these are really fit parasites in the population. And then also another important observation that we made is that the parasites which they were mainly concentrated first in the northern part of the country, but as time goes, we started seeing them in additional sites. Like in 2020, there are now much more sites. We look at the maps in 2020, you see much more sites where these parasites are detected, although we still see a higher prevalence in the northern part of the country. Then another important point I want to also highlight is the third polymorphism, which is shown on the lower maps, which is 4649F. This is a polymorphism at the same loci as the 469Y, but also plays important role in delayed parasite clearance. Over the years, it has been detected at low frequencies at different places, but 2020, we got actually intrigued when we saw this polymorphism also at very high prevalence at one site, which is in the southwestern Uganda, which you can see in the map of 2020, down on the southwestern part where you see that the prevalence was actually close to 30%. And that's pretty much prevalence, which shows that this might also be a polymorph that plays an important role in the population. So we got interested in further understanding these two polymorphisms, wanted to see whether the increase in population of these polymorphs is simply an expansion of those which emerged earlier. And they did by analyzing 7 microsatellites that flank the PFK13 gene. What you see here on the first map, of course, is the prevalence of the two polymorphisms, but on the dendrogram, you see that the parasites which are carrying the two polymorphisms closely clustered together, which might actually show or tell us that it was a single emergence that continued to increase in the population. So the key point here is that it's possible that this was a single emergency of these two populations, and there's now clonal expansion of them in the Ugandan parasite population. So the next thing that we did was that by 2020, when we observed a rapid increase in prevalence, we are concerned that we do not understand the importance or the phenotypic importance of these polymorphisms. Through some of the additional projects we have that are doing ex vivo experiments in Uganda, what we did was to set out to go to one of the sites. When you see on the first map, which is highlighted on the red side called Agago, is where we see high prevalence of these two polymorphisms. We traveled to that site, collected parasites, and transported them to another site, which is on the lower side of the map, labeled as Tororo here, where we have a cell culture lab. All those parasites were actually we transported there and undertook analysis. Specifically, we did IC50 assays and registered survival assays for all the parasites. And at the same time, there are also parasites that were continuously being collected from Tororo, and they were also undergoing the same IC50 assays and the RSA assays. So the key thing that we observed from this experiment is that, one, the parasites that we collected from the Agago site, where we are seeing high prevalence of these two polymorphisms, is that the parasites there had relatively high ring cell survival assay, which showed that one of the at least polymorphism 469Y, where we had enough number of samples, was associated with delayed artemisinin clearance. And then another intriguing observation that was unexpected was from the IC50 assays, where we observed that for one drug, which is lumefantrine, which is actually an important partner to artemether and the most commonly used ACT in Uganda, we also saw that the IC50 for the parasites from the same site, called Agago, was much, much higher than the IC50 for the parasites which are collected in a site called Tororo, where we're not seeing these genotypes. And all these parasites from the two sites were collected at the same time, assayed in the same lab, using the same people. So we think this difference was real, and we do think that there's potential that there's decreased lumefantrine activity at the same site, where we're also seeing decreased artemisinin activity. So this is therefore really much of concern to us, and we're worried what this might mean for AL efficacy in Uganda. And just to give maybe more information on what's happening in Uganda, there was also a recent study in Uganda that tried to show evidence of clinical artemisinin in Uganda. And this study was conducted in a site called Ugulu, and this site, Gulu, is in northern Uganda, where we also see high prevalence of these artemisinin resistance associated polymorphisms. And in this study, they had patients who had uncomplicated malaria, and they were treated with IV artesunate. And then they measured parasite clearance half-life, and also did ex vivo sensitivities. And what they observed was that out of the 240 patients, they had delayed parasite clearance in around 14%, which is around 5%. And out of these 14%, 13 of them were infected with the parasites that were carrying either the 469Y or 675V mutation, also highlighting the importance of these two polymorphisms to ACT efficacy. So therefore, from this story here, we do believe that artemisinin resistance has emerged in East Africa, particularly. There's evidence now that this is present in Uganda. And we believe that we have evidence that there are multiple independent emergencies of different PFK13 mutations in Uganda. And of course, there has also been now evidence that of clinical and in vitro delayed clearance after artemisinin exposure. And another intriguing observation is that we are seeing decreased lumefantrine activity in the parasites. So the key question now remains, what are the clinical implications of this? Like, has the ACT efficacy in Uganda changed? Because now if we are seeing decreased artemisinin activity and lumefantrine activity, what does this mean to the clinical efficacy of AL in Uganda? And also another key thing which is important in our mind is, will these observed parasite genotypes continue to spread in the population? So now, importantly, we also want to now start discussions on what are the measures that can be called for to limit the spread of these parasites, and also how can we mitigate the impact of this emerging resistance on malaria management. Thank you very much. I think I want to acknowledge the PRISM project team for their support in this project, and the IDRC team. That IDRC is the organization that hosts the PRISM project. Our collaborators from University of California, San Francisco, Brown University, and then also the funders, National Institute of Health that funded the ICEMR project, and also the International Society of Infectious Diseases, which gave us some small grant also to support some of the activities of this project. Thank you very much.